Hello. I'll first introduce myself. I'm Leelavati Narlikar, and I missed the first week, but I was kind of following online. Glad to be here and interact with all of you in person now. So it's my pleasure to introduce Anshul Kundaje. He is an associate professor at Stanford in computer science and genetics. And if you've at all worked on, on ENCODE data, you would have heard of him. So he's one of the key contributors, part of the massive consortium. He was instrumental in creating all the the full computational pipeline, doing the analysis, and consolidating data that was coming from many different labs. And at Stanford, he continues to work on large-scale computational genomics problems. And he's going to talk about deep learning approaches that their lab is, really working on at the forefront of in this area. So, Anshul?
Great. Let me just share my screen. And... All right, I hope you can see the screen. And yeah, firstly, thanks to Leelavati and Rahul and other organizers for inviting me to teach at this meeting. Sorry I couldn't be there in person. I have a young 10-month-old, and so we're trying to minimize travel. So it was too much of a slog to go all the way to India, although it would have been fun. So instead, my student from my lab, Surag Nair, will be there later today. He'll be landing about right now, and he'll be there in about an hour or two, hopefully. So if you want to talk more about any of the lecture or anything else, he'll be there in person. So feel free to touch base with him. He'll also be teaching, 2 live interactive collab tutorials based on the lecture I'm giving today. Today, I'm going to really sort of go through a primer on deep learning for regulated genomics. We just call it How to Train Your Dragon. And the tutorials that Surag will do tomorrow, the collab notebooks are largely based on the primer today. And then on Friday, during my research talk, I'll sort of dive more into some interesting aspects of the research that we're doing based on these kinds of techniques. Okay, and if you have any questions, feel free to stop me and ask, or hopefully we'll have some time at the end, and we can also answer any questions you have then. All right. Okay, so let's jump right in. So what we'll cover today is supervised machine learning methods for decoding regulatory DNA. I'll give a brief introduction to what regulatory DNA is, why it's relevant, why it's interesting, how we can ask questions about the properties of regulatory DNA using supervised machine learning methods. I'll talk about why convolutional neural networks in particular, a particular class of neural networks and deep learning models, are a good fit for modeling regulatory DNA. We discuss how we train these neural networks, how we evaluate them, and then we'll talk about some commonly used neural network architectures in this domain. Okay, I'm going to keep it relatively simple today. I'm not going to dive super deep into any mathematical derivation, so forth. I'm going to go through most of the high-level concepts. When the slides that you have links to other documentation, blog posts, tutorials, reviews that you can read. Hopefully, this primer will serve as a good launch point for you. You'll be able to understand those much easier after we finish this. Okay. All right. So we're going to talk about decoding genome function, and what I mean by that is, as you know, you can sequence genomes quite efficiently and accurately and cheaply nowadays. About $200 you can sequence a human genome within a day or so. And what you end up with is sort of a long sequence of A, C's, G's, and T's. There are 3 billion of these in the human genome. Sorry, let me just try to hide this video. And believe it or not, it was in 2003 that we first figured out the draft sequence of the human genome. So it's not been that many years. It's 2023 now, 20 years, right? 2 decades. very much in our lifetime, that's the first time we saw the genome, and now we can sequence genomes really cheaply. So now we don't just have a reference genome of a species, but we have actually personal genomes. We can sequence individuals, we can figure out all the genetic variation, all the differences between individuals' genomes and how they relate to phenotype. But once you sequence a genome, interesting questions pop up. So that's just the start of the story. Once you sequence the genome, you would like to know what all these A, C's, G's, and T's actually code for, right? So if there's a mutation at this position in the genome, what does it do? What does it not do? What does this particular DNA word end up doing functionally, right? What's its molecular function? What is it encoding? So this is what we'll try to answer today. And we'll use machine learning coupled with regulatory genomics experiments to try to understand these questions. Now, when you're interpreting genomes, trying to decode what genome sequences mean, you cannot do that in a vacuum. So the reason is because humans, for example, multicellular organisms, we have a whole variety of tissues and cell types in the body. Here's just a graphic cartoon of some of the core tissues and so forth that, for example, the ENCODE project, the Roadmap Epigenomics project that I'll talk about in a few slides, have tried to assay. And what's really interesting is all these cell types in the body, they actually have the exact same genome sequence in an individual. So what's actually happening is, even though there's incredible diversity of morphology and function amongst all these different cell types, somehow all of that is encoded in the same sequence. So the same sequence is interpreted differently in different cell types, giving rise to different kinds of molecular phenotypes, cellular phenotypes, and eventually organismal phenotypes, right? So when you're interpreting genomes, genomic context and cellular context both matter. By genomic context, I mean where in the genome you're looking at a word. The same word in a different place in the genome can have a very different meaning. And then the same word at the same position in the genome in a different cell type can have a very different meaning. So when you're interpreting genomes, you have to interpret them in the context of genomic position, as well as in terms of cellular context, in which cell type or condition you are studying that genome, okay? And so to understand genome function, you need to first understand like the basic anatomy of what the genome is made of. So if you take the DNA in the nucleus of a cell and then you unspool it, you will find various interesting things in it besides the sequence. The sequence is encoding for different kinds of functional elements. And everyone knows about genes. Genes code for proteins. So there are about 26,000 or so, 25 to 26,000 genes in the human genome. They are sequenced directly codes for proteins. So that process is called transcription, when you convert DNA to RNA, and then translation when you convert the RNA to protein. Proteins do many of the interesting functions in the cell. And one of the things they also do is regulate genes. So what I mean by that is some proteins actually, their key role is to control the activity of other genes, right? So the key idea is you have 26,000 genes in the genome, and then the precise function of a cell is really dependent on the precise repertoire of genes that are expressed. So which genes are expressed in what quantities and at what time really determines function, right? So there must be some recipe telling the cell which genes need to be activated and repressed. So you need a control system to control which genes are being turned on and off. And that control system also happens to be encoded in the genome. So there are little DNA sequence elements in the genome called regulatory elements. And these regulatory elements, as I've shown you here, they are segments of the genome whose job is to control gene activity. So they turn genes on, they turn genes off. Right? And if you dive deeper into the anatomy of individual regulatory elements, it's just a DNA sequence, right? It's a segment of the genome. It's A, C's, G's, and T's. But what actually is happening there is these elements, these DNA segments, they act as landing pads for a variety of important regulatory proteins, as I mentioned before. These are proteins that bind the DNA in the combinatorial fashion. And they basically, once they land and bind DNA, they can effectively either regulate genes to turn on or increase expression or turn off or decrease expression, right? So that's sort of the control mechanism, and that's called gene transcriptional regulation, okay? Now, if you want to know where all the proteins are bound in the genome in some snapshot of time, in some cell, you can in fact perform really interesting experiments, biochemical experiments using sequencing technology. These are called functional genomics experiments. And here I'm showing you one sort of, this is real data actually. These are two genes in the genome in some cell type. This gene is active, this gene is repressed, okay? And we perform an experiment called ChIP-seq, chromatin immunoprecipitation followed by sequencing. I'm not going to go into the exact biochemistry of how that's done, slightly beyond the scope of this particular tutorial. But just think of it as an experiment that gives you like signal at every position in the genome, and the intensity of the signal is proportional to the likelihood of the protein being bound at that position. So wherever you see spikes in the signal, the y-axis is sort of the strength of the signal, the x-axis is position in the genomes. And you're seeing that, you know, the large parts of the genome that don't have any signal, and then all of a sudden there's this huge spike right here, there's another spike here, another spike here. So wherever you see these spikes, those are likely positions where the protein is bound. And you can perform one experiment, and that gives you a genome-wide profile of all locations where the protein is bound. You can similarly profile other kinds of modifications or other proteins bound to DNA. So here I'm showing you something called epigenomic or histone modifications. Think of these as molecular flags tagging different types of elements. Okay, so here's an element, the promoter of a gene. The promoter of the gene is the primary element that activates the gene, that recruits the polymerase that transcribes messenger RNA from the gene. You can see that the promoter of the gene is marked by these red flags, right? When you make the measurement using the experiment, you can see kind of a nice enrichment of signal right above this promoter, right, of an active gene. You can see the repressed gene's promoter does not have any activity. Here's another marker, an orange marker, that has slightly stronger activity at these distal regulatory elements called enhancers. These regulatory elements can be far away from the genes that regulate, but typically the DNA loops in 3 dimensions. So think of this kind of DNA looping and bringing this element close to this element in 3D and regulating activity, right? So this marker is more enriched at enhancers. Here's other yellow marker. You can see it's quite nicely marking all the enhancers. It's less prevalent at the promoter. And then here's a transcribed gene marker. You can see it perfectly marks the body of this active gene. You can see the repressed gene has no signal. And then here's a repressed gene, and you can see it lacks all the active modifications, right? It doesn't have the protein bound. It doesn't have any of these red, orange, yellow, green modifications, but it has this gray modification. So you can see how the cell is using these kinds of molecular markers to actually mark different types of elements, active genes, active promoters, and the proteins that bind these promoters and enhancers, these regulatory elements. You can also experimentally figure out where they are by performing these kinds of assays. Okay, so this is really exciting because now you can perform one experiment for any marker of interest in any cell type, and you can get a genome-wide readout giving you precise positions of where these proteins are bound or these markers exist. So what NIH did actually is for almost 2 decades, since 2003, they funded 2 large consortia, one of which is called ENCODE and the other is Roadmap Epigenomics. ENCODE stands for Encyclopedia of DNA Elements. And what these consortia did is they performed these kinds of experiments. You can see hundreds of different molecular experiments, RNA expression, protein DNA binding, various kinds of epigenomic modifications, DNA methylation, you name it, hundreds of different kinds of experiments targeting different kinds of molecular events, like I showed you in the previous slide. Each of these experiments, gives you a profile across the entire genome. So one axis is a genome, 3 billion positions. The other axis is hundreds of molecular measurements in that genome. But there's a third axis, which we didn't talk about yet, which is hundreds of cell types and tissues, right? So this massive teracube of information, digital readouts from experiments. And this axis is, of course, now almost infinite because you can start doing experiments in individual cells, in single, individual cells in all individuals on the planet. So it's not anymore a reference data set for all of humanity. It's like you literally have you can generate one such teracube of information for every individual, that you get access, tissue access or DNA access to. So, these kinds of data sets are increasingly being generated, and they are really exciting because they can power machine learning models to try to understand how is the DNA sequence, the 3 billion letters, encoding all of this incredible information, how is it directing all these proteins to bind where they're supposed to bind, and how are these proteins activating genes, and then how are mutations in the DNA affecting gene activity through these regulatory elements, right? So all of these questions can hopefully be answered once we have this data and once we have built models that can help us answer these. So today we'll talk about the first part, which is really trying to use the kinds of models to understand how DNA sequence encodes regulatory activity of these regulatory elements. And then, in my talk on Friday, I'll talk a little more about how we can use the same kinds of models to predict the effects of mutations, genetic variants and mutations on molecular activity. So let's start with this piece, which is deciphering functional DNA words and their syntax in regulatory DNA. So as I showed you before, if you take any of these DNA regulatory elements, right, those little orange and red bars that I've shown you in the previous slides, I mentioned that they basically act as landing pads for various combinations of proteins. So here I'm showing you 3 proteins. They are combinatorially binding, this element. How are they recognizing this specific element? Why are they binding only this element and not other regions in the genome? But what happens is these proteins actually have DNA binding domains, and those DNA binding domains, they recognize specific fuzzy words or sequence motifs. Okay, and the DNA, through evolution, has encoded kind of various fuzzy versions of these DNA words, motifs, and combinations of these motifs exist in these elements, and these motifs actually recruit these proteins to bind there, right? Now, this sounds simple, but it's not, because these words, unlike English words, or any language words, are not deterministic. They're probabilistic. They are fuzzy. Proteins have affinity to DNA, and they can bind a whole range of sequences, some very strongly, some very weakly. So deciphering these words is actually kind of like a holy grail. There's a ton of work over decades trying to figure this out. We still cannot exactly fully model this. But more importantly, it's not just the individual words, just like in language, where you have combinations of words that can give different meaning, and then a word in the presence of other words can mean something very different. It's the same thing with DNA. Different combinations of words in different arrangements can give rise to very different downstream effects. Okay, but the key point is now with these experiments, as I mentioned before, you can create these genome-wide protein DNA binding maps. So here I'm showing you some data from ENCODE. Each row here corresponds to a different regulatory protein, like the black triangular protein I showed you before. There are thousands of these, and they all
Anshul, I think he's disconnected.
Sorry, it looks like we dropped. We try to reconnect.
Yeah.
And see where we were at.
About 1 minute, I think we lost you.
All right, can you see my slides now?
Yes, we can. That was the slide that you were on.
Okay. I don't know where I stopped, but I can.
You had said that similar to natural language, the order of the words matters and the context matters, something like that you were saying.
Okay, great. Yeah, so, like I was saying, it's not just the words that matter, the local context of the words matters as well. What other words are around it? What's the exact orientation, spacing, how many of the words are there, the precise sequence of the words, all of that matters. And I was just saying that when you perform these experiments, each row here corresponds to a different protein, and one such experiment generates a genome-wide profile for that protein. You can see these are a variety of different binding maps for several proteins in some cell type. It's a lymphoblastoid B cell in humans. You can already start seeing, really beautiful combinatorics, like right here. You can see these collections of proteins binding together at this position. Sorry, let me switch back to pointer. Okay, there we go. Right here, you see these collections of proteins binding together, right? All these spikes at the same position. If you go to a different position in the genome, you see a different collection of spikes, right? Here you see a different combination going on. So there's incredible combinatorics of binding of these proteins, and somehow the DNA sequence is encoding all of this. So we'd love to understand, decipher what the language of these regulatory DNA sequences is and how that varies across different cell types and different proteins and so forth, right? So how do we use machine learning to do this? So let's try to, the first thing you have to realize is when you're given a biological question to answer, and you're given some data, your first sort of job is to think of, if you're using machine learning to solve this problem, is to think about how you can try to convert this problem, this biological question you have, with the data you have, into a classic machine learning formulation, right? So what we're going to do is convert this question of trying to decipher, the sequence syntax of these elements into a classic supervised machine learning problem. Okay, I'll explain how we do that. So the logic is as follows: you performed one such experiment across a genome, right? So I'm showing you here some small segment of the genome. Here's the ChIP-seq experiment, which profiles binding of some transcription factor protein, the regulatory protein. It doesn't have to be that experiment. It can be any experiment, DNA methylation, gene expression, whatever it is. But you end up getting a readout at every position in the genome, right? You get some signal from the experiment. And the signal, as I mentioned before, captures some sort of notion of the likelihood of that molecular event happening at that position in the genome. So you can see there are these spikes right here, right? And so what you can do is you can take the entire DNA sequence, 3 billion positions, and you can segment it. You can bin it into little chunks, let's say 100 base pairs or 200 base pairs. And so you have millions of these 200 base pair chunks, lined across a genome. And each of these sequences, local sequences, has a corresponding readout from the experiment, right? And there are different kinds of ways you can process the data. You can actually use the signal as you measure it. So, for example, at this position, the signal is 20.2. At this position, it's 0.3. At other positions, it's even weaker. Or you can even binarize the data. You can kind of threshold it and say, okay, here's a high signal, here's a low signal, right? This is a low signal, this is a high signal. So either way, what you end up with is every bin in the genome, every 100 or 200 base pair, 1,000 base pair bin in the genome gets labeled with the molecular experiment, right? And it's either a binary label, active, inactive, strong, weak, or it's a quantitative label, like 20.2, 0.3, and so forth. And so what you end up with is basically millions of sequences across the entire genome, and each sequence has an associated label from the experiment, right? So you can actually think of building a supervised machine learning model whose goal is to take some inputs, in this case DNA sequences, and learn a mathematical mapping from these inputs to labels, right? So sequences to labels. This is a classic classification or regression task, right? In classification tasks, you're taking some kind of inputs and you're trying to map them to some kind of discrete labels. In this case, they're looking at binary labels. They need not be binary. They could be multi-level labels. You could say like very high, high, somewhat low, very low, right? You can have multiple classes. Or it can be a regression task where the goal is to map the same inputs to quantitative values, okay? And there's a whole bunch of literature in this space. You can apply all kinds of models, linear models, you can apply support vector machines, you can apply neural networks. Today we'll talk about how we use neural networks, okay? So what does a supervised machine learning model do? Well, it takes some input, it pushes it through a series of transformations, and those transformations are basically performed by taking the input and applying some kind of adjustment, to the input. Usually there are some parameters or weights, and you learn these weights. That's part of the learning process. But basically you take the inputs, you transform them in some way using the weights or the parameters of the model. And sometimes some of these models have multiple layers. So you pass on these transformed versions of the data to additional layers of transformations. The parameters are used to perform these transformations, and eventually, once you finish all these transformations, you get an output, right? The predictions of the model. And you hope that the predictions of the model are as close to the true targets, target predictions, target labels as possible, right? That would be a good model. A good model would make predictions that are very similar to the true targets. How do you determine how similar these are? You use something called a loss function. The loss function compares the true targets and the predictions and computes a loss score. And your goal is to learn the weights of the model. And usually this is done, in various using various algorithms. One of them is an iterative strategy where you have an optimizer, and the optimizer basically, takes the predictions of the model, looks at how similar or different it is from the true labels, and then it tries to update the weights of the model to try to make the predictions as close to the labels as possible. So the key thing you need is a model definition, some kind of parameterization of the model. You need some loss function to tell you how different the targets are from the predictions. And then you need some kind of algorithm or optimizer that takes the loss function and then uses it to update the weights of the model to make the predictions as close to the targets as possible. Okay, that's classical supervised learning. So the important question to realize is, if you're given a DNA sequence, how do you actually represent it, right? And typically what you do is you take sequences and you'll featureize them in some way. You'll convert them into a table of features, right? So you have millions of sequences across the genome. They have labels. Each of them has a label of 1 or -1. Let's say binary for now. You can take these sequences and you can, for example, one kind of featureization is a bag of k-mers, like a bag of words model. And here the idea is you can take the sequences and you can just enumerate all possible k-mers of size 3, right? So AGA, GAT, ATC, all possible k-mers of size 3, and simply count how many of these k-mers you find in each sequence, right? So that's an interesting featureization. So now you have this millions of sequences and you have a fixed number of features. And then you're trying to learn a mapping from these this vector of k-mer counts to binary labels, right? Let's talk about some models. So the first model we talk about is a linear regression. What linear regression does is takes these bunch of inputs, the input features, right? These k-mers, for example. It applies some weights to them, just multiplies the k-mer counts with some weights, adds them up, linear combination, and there's a bias term right here. And that's it. That's the output, right? Very simple. This is a linear regression model. You can also call it a linear artificial neuron. And training this model means, or training this neuron means learning the weights, right? Learning these parameters of the model. And again, there are many different algorithms to learn parameters of linear models. Most of these are closed form equations. You don't actually have to do some kind of iterative algorithm. You can just directly solve it and obtain the parameters of the model given the data. Okay. Now this is a very simple linear model. You can make it slightly more complicated by taking this simple linear model that you had before and then taking the output of the linear model and then pushing it through a nonlinearity. And one nonlinearity that's very commonly used for binary outputs is a logistic nonlinearity or a sigmoid. What the logistic or sigmoid does is it takes the output of this linear combination of the features, right? And remember the Z, right? The linear combination can be from -infinity to infinity, right? It can be across the entire real line. And the sigmoid or logistic function compresses this in the range of 0 to 1. right? And the equation is basically 1 divided by 1 plus e to the negative z, right? So this is what the function looks like. As you can see, z goes from minus infinity to infinity, but the logistic function, the output of gz is always between 0 and 1, and it saturates, right? After some values, it saturates to 1. After some values, it saturates to 0. So your final prediction is now doing a linear combination of the features and then pushing it through. Something happened here. And then pushing it through a logistic function, right? Okay, and this is basically logistic regression. So a simple nonlinear sigmoidal neuron is performing logistic regression. Again, training this model means providing some labels and some inputs, and then trying to learn these thetas, the parameters of the model, given the data. And again, for logistic regression, there are many simple algorithms to infer these parameters. Right? Now, let's think about how we can construct a deep neural network from these two basic concepts. A deep neural network is just a concatenation of multiple layers of simple, combinations of neurons. Okay, so this input layer is again the features of each sequence. So here's one sequence. It has some features. It has various k-mers of size 3. You have the counts of these k-mers, right? Those are the features. So let's say this has, this sequence had 3 AGAs and 0 GATs and 1 ATC, right? So that's the input to the model. Think of that as the input layer. And now in the first hidden layer of this neural network, you'll have exactly the neurons I showed you before, right? Think of imagine having logistic neurons. So you have this is the output of this neuron, and it takes these 3 inputs and combines them in some linear way through some weights. Each of the arrows correspond to different weights, parameters, right? Those thetas that I showed you before. When instead of having just one output neuron, like I showed you before right here, there's just one output, right, from the 3 inputs. In this case, you have multiple neurons. Each of them is using the same 3 inputs, but generates different outputs. And why does it do that? Because they have different weights. We've not yet talked about how we learn those weights, but just assume we have those weights, right? So now we have 4 neurons right here. Each of them is a logistic model, for example. Each of them takes these 3 inputs and produces a different output. Now, we don't stop there. We push the outputs of these 4 neurons to 4 other neurons in the next layer. And all of those 4 neurons now operate on the outputs of these 4 neurons as inputs. Again, perform some linear combination, push them through a sigmoid and produce an output. And you can keep doing this. You can keep passing them through multiple layers of nonlinear neurons, which take linear combinations of the inputs from the previous layer, perform a nonlinear operation, and keep going, right? And the final output layer of the model is basically logistic regression. It's just taking the final layer, taking the outputs, doing a linear combination, pushing it through a sigmoid, and predicting, some probability between 0 and 1, right? So can somebody guess how many parameters does this neural network have? I know you have my slides, so you shouldn't cheat. You should just tell me what it is.
Any answers? 40 from somebody.
Okay.
Anyone else? Sorry? 32?
All right, so this is the answer, and I'll tell you how we got to that. So there are 3 inputs. There are 4 neurons right here. There are 4 times 3, 12 weights here. Now, these 4 neurons in the next layer take these 4 inputs. So it's 4 times 4, right? 16. This neuron takes 4 inputs, right? So there are 4 weights here. So it's basically 3 times 4 plus 4 times 4 plus 4 times 1. And then there's plus 9, right? That's the odd thing. Where did this plus 9 come from? Well, remember that each of these neurons has this bias term, right? The bias term is it's an offset, right? It's not multiplied against any input feature. It's just a constant, but that's also logged, okay? And that's an offset. So each of these neurons actually has an offset. And there is 1, 2, 3, 4, 5, 6, 7, 8, 9 neurons. Each of those neurons has their inputs with their associated parameters, but also has these offset parameters, right? So in total, there are 9 additional bias terms or offsets. And so you end up with basically 41. Okay. So when you talk about these neural networks, as you can see, there are many interesting choices to make here. Why did I choose 4 neurons in this layer? I could have chosen 5, I could have chosen 10, I could have chosen 100. That's choice. Why did I only choose 2 layers? I could have chosen 20 layers, right? So all these are called hyperparameters. Because they're not parameters of the network, you have to pre-choose them. And once you choose these hyperparameters, these architectural properties of the network, then you have set the number of parameters, and your goal is to learn those parameters, right? So typically, there are these additional decisions you have to make about what exact architecture to use. And so when you train neural networks, you're not only trying to learn the parameters of the network, but you're also trying to learn what is the optimal architecture, what's the optimal hyperparameters of that network itself. So there's 2 loops of learning, right? One is an outer loop, which is sort of what's the hyperparameters, and then there's, given the hyperparameters, what are the parameters? Okay, so both are very important. Also, we talked about one kind of activation function on nonlinearity, which was the sigmoid or logistic. There are many others. There's a tanh, there's a ReLU. ReLU is a rectified linear unit. This is a very important one, which is commonly used. It's a very simple function. It's a function that's linear as long as the input is positive or non-zero. But if the input is negative, basically the output is thresholded to 0. Okay, so it's basically a max function between 0 and x. Okay, so if x is greater than 0, it's x. If the input is less than 0, it's clamped to x. So it's a thresholding function. Okay, there's tanh. There's other kinds of these kinds of locally linear nonlinearities. There's PReLU. There's a max out. There's soft pluses. There are hundreds of these functions that people have built. Okay, and they all have different properties, and they all have different useful aspects. Again, I don't have time to discuss why each of these are important, but let's just focus on these 2 for today: the ReLU and the sigmoid. And usually, even today, most of the models typically use some version of a ReLU, like a soft plus or ELU or something like that. Okay. All right. So how do we train these models, and how do we learn the weights of the neurons? So I mentioned before that you remember my little cartoon of supervised learning. You need a loss function, right? The loss function is a measure of how similar the predictions of the model are to the actual observations. So let's say the observations, the actual labels on the sequences are labeled yi. And the predictions are h of xi, right? And theta are the parameters you're trying to learn. If you're trying to do a regression, that where your y's are real valued, right, not binary, typically you'll use something called a squared loss. The squared loss is very simple. You just subtract the prediction of the model from the actual label. You square it, so you get rid of the sign effect, because you could be greater than or less than the label. Just basically you're sort of taking the square of the difference between the observation and the prediction, and then you sum this over all the examples, all the sequences in the data set. You divide by half. This is just a normalization. This is a constant. It doesn't really matter in the end. But basically, it's just a sum of the squares, right? Or squared loss between observed and predicted labels. Okay. Very simple function, right? Quite nice. It's very commonly used for regression. For binary classification, you typically use the logistic loss, which is the exact same loss that logistic regression uses. Or it's also called the binary cross entropy. In this case, what you're doing is you're taking the labels and you're computing the entropy, cross entropy between the labels and the predicted property distribution, right? So you just take, for example, yi log of hxi. Hxi is in this case, the output of the model is a probability between 0 and 1. So you're basically multiplying, the label times a log of the probability plus 1 minus y times log 1 minus h. Right. So if the label was 1, then this is the loss. The label was 0, binary prediction. This term cancels out. Yi is 0, but 1 minus yi activates, and then it's log of 1 minus h. So, and then you again sum this over all the examples, and you take the negative. Okay. So this is a negative. It's the binary cross entropy or the logistic loss. And the goal is to minimize this, right? You want to minimize this error and you want to minimize this error. Okay. So how do you actually minimize this error? Now, as I said before, for simple linear models, which have just, no hidden layers, you can literally just take the derivative of this loss and you get a closed form equation for every theta, okay? But when you have this kind of deep neural networks with many, many layers, you can't do that. You need, you can't solve it explicitly. You need kind of an iterative algorithm to solve it. And one of those algorithms is very famous, called stochastic gradient descent. The idea here is that you define some loss function, j of theta. These are the two loss functions that I just defined. The goal is to optimize, this loss, minimize this loss. You want to get to the minimum possible loss, right? That's good for you. Ideally, it should be 0, but maybe you won't get to 0. You'll get to some very low value. You want to find the thetas for which this loss would be very, very low. So you define a loss function. We defined it in the previous equations. And now you want to get to the minimum of this loss. Hopefully, this loss is differentiable, in which case what you can do is imagine like a very simple example of only one parameter, right? A neural network having just one parameter. A loss function would look somewhat like this. This is like a convex loss function, right? It's differentiable and convex. And you start by just randomly initializing the weights of the model, the parameters of the model. You just make some random guesses. So you could be anywhere in this on this x-axis, right? You just randomly. So I initially randomly guessed this value right here, this theta. And for this value of theta, I'll compute the loss, the j. So what would happen is I'd be on this curve, right? This is the loss curve. And for this value of theta, the loss would be this value, right? The j of theta. Now, what I want to do is, starting from this position, I want to get to the minimum, right? So how do I get to the minimum? What's the direction of the steepest descent, right? If this was a hill and I was climbing down the hill and I want to know what is the direction I need to walk, right, down this surface, what is the best possible direction I could walk in? The answer is if I take this point where I currently am at and I draw a tangent, right, to this loss curve, the tangent actually would give me the precise direction, the direction of steepest descent, right? And so it gives me a nice way, like, this tangent basically is the slope at this position, and that is precisely the gradient of this loss function, or the partial derivative of this loss function with respect to this parameter, gives me exactly what I want. I want the slope, right? I want the direction of greatest descent. So what we will do is compute the gradient, right, of the loss function. And then we will walk in the opposite direction. The gradient points upwards. We're going to go in the opposite direction, right, of the gradient. So what we do is take the current, we'll take the current theta, and then we'll take the current value of theta, and we'll take a small step in the direction opposite to the gradient, right? So we'll walk downwards a small step. We don't take too large a step because then we'll kind of move somewhere really far away. Take a small step. The size of the step is controlled by this parameter alpha. That's called the learning rate. And the direction of the step is effectively this gradient, right, of the loss function with respect to that theta, okay, which is just a slope, right? It's just giving you the direction of descent. And you keep doing this. You take a small step, you get to the next position, you compute the loss at that position for that new value of theta, you get the gradient at that position, and you take another small step, and you keep going. And if you keep doing that, you'll eventually hit rock bottom. And when you hit rock bottom, the gradient, the slope of the tangent will be 0, right? So at that point, your thetas will not update anymore because the gradient becomes 0 and your thetas become stationary, right? So to get to the bottom of this pit, you need to take these small steps in the direction, in the opposite direction of the gradient, right? The gradients are just giving you the slope. Now this is in 1 dimension. Now imagine what happens in 2, 3, 4, 5 millions of dimensions, right? You can't visualize it, but it's the same idea. In high dimensional space, you're trying to walk down some high dimensional surface, and you're trying to get to the minimum of the surface. The surface is not going to be as beautifully simple as this. It's often going to be a very complex surface with hills and valleys and tall mountains and deep valleys. And you want to get to the absolute bottommost valley in that entire landscape, or all possible values of all parameters, right? So the stochastic gradient descent is a very powerful algorithm that will allow you to get that, right? So as I mentioned before, this is called the gradient, and alpha is called the learning rate, which tells you the learning rate, how quickly you can descend. Okay. And now so the key thing to do is, if you're trying to get to the bottom of this pit, the key calculation you have to figure out is computing the derivative, right, of the loss function with respect to every parameter. Now that is not trivial either, because you can have a parameter somewhere deep in the model, and the loss function is in the output, right? You somehow compute this gradient very efficiently from the output of the output loss function with respect to each of these parameters. So here what we can do is use another powerful algorithm called backpropagation, which is basically chain rule of derivatives, right? So if you want the derivative of z with respect to x, and you have a y in the middle, x is passing through y is a function of x, z is a function of y. You can easily do that. You can use the chain rule, where basically, dz by dx is equal to dz by dy, which is easy to compute because these are directly connected. Z is a function of y, so you can often write down, and most of the neurons we saw are very simple, right? They're just computing some linear combination of the inputs and then pushing them to non-linearity. It's basically like a logistic model. It's very easy to compute the local gradient, of dz with respect to the input that was given to z, right? So the dz by dy and the dy by dx are very easy to compute because they're all simple local linear logistic models, right? But you want to get from z to x, and x could be 10 layers down and many different connections. So to do that, you just compute all these local gradients, and then you use chain rule, right? So dz by dx equal to dz by dy times dy by dx. And sometimes you will not have just one linear path. You will have often multiple paths going from x to z. And there you can again use additional properties of derivatives, where, for example, you're taking the sum of the chain rule across all the possible paths from x to z, right? So dz by dx would be dz by dy1 times dy1 by dx plus dz by dy2 times dy2 by dx, right? And so if you have a very large number of paths, it's very simple. It's just like a sum over all possible paths of the local gradients multiplied together through chain rule, okay? And that basically, this can be implemented very efficiently using dynamic programming. And that's basically called the backpropagation algorithm, okay? So the backpropagation algorithm is used to compute the gradients of the loss function with respect to every parameter. And then you just update the parameters of the model using the gradients. And here what I'm doing is I'm literally computing the gradient of the loss function with respect to the parameter for every example, right? So this is example xi and x and yi. It's called stochastic gradient descent because you're randomly sampling examples, okay? By examples, I mean inputs, xi, yi combinations. You're computing a gradient for every single random example, and you're making small steps. You can also do something called mini batch gradient descent. When instead of doing this for every example, you do it for small batches of examples. You randomly sample some batch of examples, you compute all the gradients, and you sum them up, and you do one step. Okay, so that's called mini batch stochastic gradient descent. And you can do batch gradient descent, which would be if you sum these gradients over all the examples in your training set, and you made a step, you updated your parameters, that would be called batch gradient descent. Mini batch gradient descent is quite powerful because it's a good trade-off between using one example at a time, where you're going to make very small steps and potentially also suboptimal steps. With mini batch gradient descent, you're taking small batches of examples. And so you don't need to actually compute the gradient over all your training examples. And often you'll have millions of these. You can just take 100 or 200 examples. You can compute the gradients for them. You can sum them up and make local steps, right? And these are guaranteed to converge to at least some local optimum, if not global optimum, okay? So that's the basic algorithm is used to train all neural networks, okay? So now when you're visualizing the loss learning curve, by what I mean by this is, as you're training the model, you're giving it all these examples, right? You're giving it one example at a time, or little mini batches sampled over the training set. And each of those passes through the data set. You're given some millions of sequences, and you're going to sample small batches of sequences. You're going to pass through the entire data set through small batches of sequences. and you're going to do that again and again. Each such pass through the data set is called an epoch. And these are typically called learning curves. So what you're doing here is as you're training the model across epochs, you're plotting the global loss. By global loss, I mean the sum over all the examples, loss per example, right? And if you're learning well, your learning curve should look like this, right? It should be decreasing kind of smoothly, and eventually it should asymptote, right? And this is good. This means your loss is decreasing as you should expect, and then it clamps down. This means you finish learning. Remember the parameter alpha, which was the learning rate, which tells you the size of the steps you're taking. If that's set too high, you're going to kind of diverge. Why? Because if you go back here, instead of reaching the minimum, if you're taking very large steps, right, you kind of hop, on the left and the right side of the minimum, right? You'll never reach the exact minimum because your step is too big. As you're getting close, you'll make a big step and jump all the way here, and then you'll jump here and you'll jump here. You kind of randomly jump around because your alpha is too large. If your alpha is too small, a low learning rate, you will learn, but you'll learn very slow. If you have a high learning rate, you might actually clamp to some kind of local optimum instead of clamping to a likely global optimum. So when you're training these models, it's very important to actually plot these loss curves and see what your loss curves look like. And they should kind of look this way, kind of smooth, decreasing, and then eventually asymptoting. So this is a very important thing to do when you're training these kinds of models. Okay. So how do you measure predictive performance? Well, once you train the model, you want to actually measure how it's doing. So typically what you do is, for a binary model, as I mentioned before, it's going to predict some output within the range of 0 to 1, right? So you can pick some threshold to decide whether the output, based on the threshold, you can say, if my output is greater than this threshold, I'll predict positive. If it's less than the threshold, I'll predict negative. And so let's say I pick a threshold of 0.5, right? The model is predicting values between 0 and 1. So let's say I put a threshold of 0.5, and I say if it's greater than 0.5, I'll predict positive. If it's negative, if it's less than 0.5, I'll predict negative. So now I have some threshold, and I can create a contingency table where I have actual labels and predicted labels, right? On actual labels on this axis, predicted labels on this axis. And I can look at, how many positives did I get correct, right? So those are my true positives. How many positives did I predict as negatives, right? Those would be false positives, right? Sorry, how many actual negatives did I predict as positives? Those would be false positives. How many actual positives did I predict as negatives? Those would be false negatives. Then how many actual negatives did I predict as negatives? Those would be true negatives, right? And so from these 4 numbers, I can compute many metrics of performance. I can compute what's called sensitivity, which is the true positive rate. It's also called recall. I can compute the false positive rate. The opposite of that, 1 minus the false positive rate, is the specificity. I can compute the precision and the recall, right? The precision is the true positives divided by the total number of predicted positives. And the false discovery rate is 1 minus precision, which is false positives divided by the total number of predicted positives, right? And it's pretty important here to distinguish between FDR and FPR. People often get confused with this. The false positive rate is the same number, is the false positives divided by the total number of actual negatives, right? The false discovery rate is, again, the numerator is the same. It's the number of false positives. But in this case, you're dividing it by the number of predicted positives. So it's the proportion of predicted positives that were false positives, whereas the false positive rate is a proportion of actual negatives that are false positives, okay? So this is for a specific threshold, right? And, but you don't know what the threshold is. Like, you can easily adjust the threshold and you would get kind of different tables, right? So to really understand the performance of the model, you'll actually do this over all possible thresholds. For every possible threshold, you will compute this table and you'll compute all these metrics. And you can plot what's called a ROC curve, or a precision recall curve. The ROC curve basically compares the true positive rate of the model against the false positive rate for all possible thresholds. So there are various points on this curve, right? And a perfect classifier would have actually 0 false positive rate and a perfect true positive rate, right? So it would kind of like this curve or all the possible thresholds would kind of hug the y-axis and the x-axis. So the area under this curve would be 1, right? The area that if you kind of shaded the area under this curve, it would be 1. So perfect classifier would have area under the ROC curve of 1. A random classifier would have an area under the ROC curve of 0.5. Okay. So that's sort of what the ROC curve looks like. You can also compute a precision recall curve. In this case, the recall is the same as the true positive rate, but the precision is 1 minus the false discovery rate, whereas here we're looking at the false positive rate, right? And again, it's a trade-off between the two. As you change your thresholds, you're either going to get very high recall with low precision, or low precision with high recall. So the ROC curve and the precision recall curve allow you to trade off between different properties of the data, right? How well you predict true positives versus false positives, or true positives versus false discoveries, right? All right, so let me see how much time I have. It looks like we are almost out of time. So I'll have Surag continue where we left off. But let me just finish 1 or 2 other slides, and then I'll let you go. So the AUROC can be quite misleading actually for imbalanced classes. And this is very important because if you go back to some of the examples I showed you, right? For the genomic sequences examples that I showed you, they're coming from, the data set is coming from an experiment, and the experiment is measuring where these proteins bind, right? So the positive labels were the positions where the protein binds, and the negative labels were all the other positions in the genome. Now, most of these kinds of events on the genome, biological events in the genome, are very sparse. What I mean by that is there are at most like, a few thousand events, binding events in the genome. So the genome is 3 billion base pairs, and there are millions of, sequence bins, and only about, at most, 10,000, 100,000 of those bins will be associated with a positive label. So when that happens, you have immense class imbalance. What that means is you have a much bigger number of negative examples than positive examples, negatively labeled examples and positively labeled examples. And when that happens, the area under the ROC curve, which is often used to capture performance of a model or all possible thresholds, can be quite misleading. I'll try to explain to you how this works. So here I'm showing you 2 examples. In this example, the data is quite balanced. So these blue lines you're seeing, they correspond to positively labeled examples, and the gray lines you're seeing correspond to negatively labeled examples. And you can see there are about equal number of blue lines and gray lines. Now, what I'm showing you is a classifier that happened to score these examples with different scores between 0 and 1. And the highest scoring examples are on the left, and the lowest scoring examples on the right, right? So in this particular classifier, you can see what happened is that most of the top scoring examples are blue, which is great, right? So what's happening is the blue examples, the positive examples, are universally ahead of the negative examples, right? And this is a good classifier, right? Because that's what you want. You want most of the positive examples to be ahead of the negative examples. So if you're making predictions, and then you are doing some kind of a downstream experiment, or you're using the predictions for some, I don't know, for some downstream, analysis, as you went down the rank list of predictions, this is the topmost prediction, and the next one. You're seeing that as you're going down the rank list of predictions, you're getting all your positives, right, mostly correct. You're getting a few negatives kind of interspersed. It's about 1, 2, 3, 4 negatives interspersed within a big sea of positives. That's great, right? That means your classifier is doing a really good job ranking most of your positive labeled examples above your negative labeled examples. And so the area under the ROC curve is very high, and the area under the precision recall curve is also very high, telling you that this is a very good classifier. Now let's take another example. This is a different data set. This is a very imbalanced data set. As you can see, there are only 5 positively labeled examples, and there are several hundred negatively labeled examples. And now what you're seeing is, again, on average, the positives are quite a bit ranked higher than the negatives, right? They're much more to the left. All the blues are much more to the left than, to the right. So there's an enrichment of the positive examples in the higher ranks. But notice what happens that as I start going down the rank list, right, the topmost example I picked with the highest score is a positive. And then I have two negatives. And then I have another positive. And then I have two negatives, another positive and two negatives. So what's happening is as I'm going down the rank list, I'm having a huge number of false positives show up. So for every true positive that I pick, I'm finding two false positives right next door, right? So this is actually a pretty bad classifier, because if my goal was to, if I knew that I was looking for like 5 positive examples, I would use my classifier to make predictions, and then I would take the top 5, take 1, 2, 3, 4, 5, and I just get 2 of them right? The remaining all would be false positives. Now, when you look at the area under the ROC curve, or you plot the ROC curve, it actually looks pretty good, right? The area looks, this ROC curve and this ROC curve look nearly identical. The reason is because the area under the ROC curve, actually what it's measuring is the probability that a randomly selected positive is ranked higher than a randomly selected negative example. And because the class imbalance is very skewed towards negatives, on average, yes, an average positive is ranked higher than an average negative. But the precision recall curve is looking much more at whether the collection of positives together are ranked higher than the collection of negatives, right? And when you're actually using these predictions in a real setting, you care about, as you go down the rank list, how many positives and negatives are you actually picking up, right? So you can see that for the same ROC curve, the precision recall curves tell very different stories for these 2 data sets and 2 classifiers, right? If I just looked at the area under the ROC curve, I would say this classifier is really good. This classifier is also really good. But then the precision recall curves tell me that this classifier is actually truly really good, because as I go down the rank list of predictions, I mostly get positives correct, right? Whereas this one, my false discovery rate is very high, because amongst my predicted positives, if I took the top 20, right, the proportion of false positives within those predicted positives is actually very high. So my false discovery rate is actually really quite low. So often you'll see in genomics papers, or in many papers actually in the literature, most problems in real life actually have this class imbalance. Most of the events you're looking for, you're trying to predict, are sparse. And if you just report the area under the ROC curve, your numbers can look really nice, 0.95 or whatever. But in reality, it's not going to hold up, right? It's going to be pretty bad. So you really need to look at both these curves and make sure that the false discovery rates are also being controlled for, and the precision recall is just as good as the ROCs. Okay. All right. So let's see. We are at 10:06. I'm already over time. So I'm going to pause there. Do we have any questions?
Yeah. Any questions? There's one there.
Hello, Professor. So
Yes.
last slide you were showing about class imbalances, right? So in case A, where we had very few positives in comparison to the negatives, how would you suggest we move forward? Would you rather say we trim the data to the top 10 and then move forward with an analysis, or what would you suggest?
In this, you mean in the first example case?
Yes, in case A.
In case A, actually, you actually just have a pretty bad classifier, right? Like you have trouble using this classifier in real life. So for example, imagine we weren't doing this for sequences and we're actually predicting whether a patient has disease or not, right? And you take your patients and you rank them based on the score of the classifier. And this would be a pretty bad clinical predictor, right? Because for every 2 patients that you predict correctly, you have 2 patients that you're making false predictions for. So this kind of a classifier would be kind of a, like a no-go, right? For real life setting. You can't rescue this kind of a classifier. But the key point is actually in many medical prediction scenarios, clinical prediction scenarios, and genomic prediction scenarios, in papers, people report the area under the ROC curve, right? And even for this classifier, the area under the ROC curve will look really good. It will be in the 0.9s, 0.95s. So it sounds really great. VCs will fund your startup, but in reality, that classifier is not going to be particularly useful. And believe it or not, people still are doing this all the time, intentionally or unintentionally, especially if you want to sell your paper to a big journal, in your abstract, it's nice to say my ROC, area under the ROC curve was 0.93, right? That's awesome. But then actually in reality, this is not a usable classifier. So there's nothing you can do to rescue this. There really isn't, right? You just have a really high false positive rate. If you can, false discovery rate. If you can withstand that high level of false discoveries, then you're okay. Like you probably have to do, twice the number of experiments to pick up all the positives, right? In this case, like you're gonna have to do close to, 20 experiments to pick up the 5 positive labeled examples, right? So that's not a particularly good classifier. Nothing you can do to rescue this. But all I'm saying here is that using both metrics is very important because it tells you, the reality of the situation as to how good your classifier is for real world setting, where you care about false discoveries, not just false positive rates. Does that make sense?
Hi.
Yes.
Nice talk. I just had a follow-up question.
Yes.
So you talked about balanced data set. And my question was, in case of imbalanced data set, when I use the oversampling technique like SMART, I can see that the data is still biased towards the majority class. Can you comment on this?
Yeah, so for neural networks, actually, there are 2 strategies that you can use. So let me just okay, can you yeah. So when you're training neural networks, as I mentioned, you have these mini batches of examples, right? So just oversampling positive examples overall need not necessarily help you, because what can happen is your mini batches, right? The little batches of examples you're picking to train the model. You can still have series of batches which just have all negative examples, right? Because negative examples are much more prevalent. So what you really want to do is try to not just globally upsample your positives, but within each mini batch, try to guarantee some balance between the positives and negatives. And that tends to work much better than either upweighting, the positive examples or globally upsampling. Does that make sense? So within each mini batch, within each little chunk of examples that you're selecting, you use, for example, let's say you had a 100:1 ratio of negatives to positives. You can try upsampling your positives in a uniform way. So within each mini batch, you have, let's say, 1:10 ratio or a 1:1 ratio of positive example and positive and negative examples, as compared to the 1:100 that you actually had in your data set, right? And that will allow your model to learn better. But then what's going to happen is the model is going to behave like as if the data was balanced. So what that's going to do is it's going to create an uncalibrated classifier. What that means is the predictions of the classifier aren't really reflecting the actual probabilities of the events. And so what you have to do then is you have to do a recalibration step at the end of the training. Once you train the model, you recalibrate it, and there are various techniques to do that. But that would be the effective strategy for. If you have highly unbalanced data, you balance your mini batches. So you try to reduce the imbalance in your mini batches. You guarantee that by upsampling your positives. You train the model, and then you recalibrate it so that the probabilities are actually capturing the true expected probabilities. That's what you want.
If anyone online has questions, can you raise your hand and ask? Anybody offline? Any more questions? So we are kind of 10 minutes into the coffee break. So let's thank Anshul again, and Sorag will take over from him tomorrow. Thanks, Anshul.
All right, great. I'll talk to you again on Friday. I might join tomorrow as well for some time, but if not, Sorag will take over, and then we can continue this discussion then.